Hello and welcome to part 2 of Chapter 19, Descent with Modification. So we've been talking about this idea that evolution is supported overwhelmingly by all this evidence. We've talked about direct observation at length, specifically with natural selection. Let's talk about these other 3 categories of data now, starting with homology. And we'll start with homology from an anatomical perspective, and specifically homologous structures, therefore. And so a great example of homologous structures would be the forelimbs of vertebrate animals, or many groups of vertebrate animals. And we can see examples of that here with mammals, turtle, human, horse, bat, and seal. We also have a bird in here as well. And if you look at the bone structure, we see the same repeating pattern here. It's colored for your understanding, where we have a single bone, 2 bones, a bunch of little bones here, and then these long bones that in humans make fingers, and other animals similar, or say in a bat, these kind of structure for the webbing of the wing here. But we see those same bones. So for a limb itself, could be considered a homologous structure. And then each of these individual bones would be considered homologous to one another. And so a critical point about homology here, not just that, it has the same bones or whatever, but that these are structures that are derived from the same body part in a common ancestor. They have a common evolutionary history or common ancestry. Another cool place to look at these anatomical homologies is in embryology or early development. And we see a lot of really tight connections here in early development. And if you think back to an earlier chapter where we talked about using, say, fruit flies or fish as models for development in other organisms, including humans, how can that work? It's because some of that early development with fruit flies may be more at a genetic level, with fish, genetic and at a morphological level, because they are also vertebrates. We see these similarities between organisms in early development. A great example of this are pharyngeal pouches. And so this is a structure that vertebrates possessed. In humans, these ultimately develop into glands and ducts, but in fish, these develop into gill slits. So we can see some examples of different organisms here. This adds on tail too, highlighted in green. Here's a human embryo, chicken, snake, salamander, fish. You see the snake tail, obviously much longer, but all these organisms have developmentally this structure that could be a tail. In humans, that doesn't ultimately develop into a tail. But here in red highlighted are those pharyngeal pouches. And so we see these developmental similarities. Here we can look at it in a different way, pharyngeal arches in human versus, say, a shark, so a fish. These in the shark are going to form those gill arches. In the humans, what do those form? Glands and ducts. And we can track the development of these structures from these pharyngeal pouches, forming these different cartilaginous or bony components of human anatomy, ear bones, throat, these tracks of where the nerves run, different muscles. They sort of follow these same patterns. Ultimately, obviously, they're not forming gill arches, but they do form these different structures in, say, mammals. Another kind of structure that we might see when we're thinking about homology are vestigial structures. And this is always a student favorite. Vestigial structures have no apparent function. And that's critical, that idea of apparent, because what's really happening here, what's underlying and what's more important is that they've lost their ancestral function. Maybe they do have some function, but they've lost their ancestral function. So a great example here would be hip bones in a whale. These whales do not have hind legs. What is the point of having hip bones? We know now that the hip bones actually, for whales that possess them, actually might provide some support for reproductive structures in males. And then if we look at some of these other examples here, humans, appendix might be important for in the immune system. boa constrictors have these rudimentary hind legs. So they have hip bones and these little nubbins here that were legs in their ancestors. Lost the ancestral function, maybe has some function today. Probably the more clear-cut example of a vestigial structure would be blind cavefish. So they have non-functional eyes, and really that structure has absolutely no function that you might conceive of. All right, so what must be true of an organ that's described as vestigial? All right, yeah, the key there is it's still homologous. So it has the same ancestral origin as some other structure in an ancestor, but it's lost its function, or it now has maybe a different function, but apparently has lost its function. Now, thinking back broadly about homologous structures in general, it's really important not to confuse homology with convergence or analogy. And the easy example of this would be flight in different organisms. So flight has evolved, say, in insects, for example. Here are the wings on insects. Wings in insects are not homologous to wings, say, in birds. They have a different, distinct evolutionary origin. Same for, say, this flying fish, right? This has a different origin than wings in insects. And so when we see these kinds of features evolve independently, this is called convergence, and it kind of makes sense, right? You're converging on a similar form from sort of two different starting points. And these are not homologous features, but they're analogous features or analogs. And so here's just yet another example. So this sort of streamlined body form in these different organisms. Here we have two different kinds of fish. For example, maybe those could be homologous, although maybe there's different fish species kind of in between that don't have those same structures. But we see these forms develop in these different organisms that live in a similar habitat. So that's really important, is we might see convergence given similar environmental conditions. And it's important maybe these are obvious examples, but it's important sometimes when these examples of convergence are subtle that you're not confusing that with true homology, which you might be using to understand evolutionary relationships. These are not going to tell you anything about those evolutionary relationships. It's just more of an artifact that these have similar forms, but again, they're different origins. Great examples of this would be in mammals, so marsupial and placental mammals. Think marsupials, Australia, right? Placental mammals like us, and you see these different groups of mammals, but similar forms given similar environmental conditions. So we can look at placental versus marsupial mammals here. We have a burrowing form, this mole, marsupial mole, and a mole. A nocturnal insectivore, climbing animals. So these are all examples of convergence. All right, so a question for you. Ancestral birds and ancestral mammals have a common ancestor, okay? But today, penguins, which we know are birds, seals, they're mammals, they have forelimbs adapted for swimming. All right, so what best describes the relationship of the bones in those forelimbs of penguins and seals? And then what term best describes the flippers of penguins and seals? So there's really two parts to this as you consider your options. All right, yeah, so the bones themselves are homologous because they have a common evolutionary origin, but the idea that they have evolved into swimming structures, that's happened independently in birds, say penguins, and in mammals, these seals. So those are analogous as swimming structures. Another place we can look at homology is at the genetic level. So this could be really useful looking, say, at genes that are shared between different organisms. And again, organisms are fairly closely related. They share a lot of genes. There are certain genes that every living thing has. Remember, every living thing has ribosomes, right? So every living thing has ribosomal DNA to code for that RNA that makes up those ribosomes. So that would be an example of a gene you could look at in any living thing and make comparisons. And here's the general concept. So we have a gene from this ancestral species, and then we have the population split somehow. I'm saying dispersal to a new island here. And, over time, DNA replication, some mutations might occur. And so in this population, this thymine crops up, substitution, and then this population, an adenine crops up over time. And because there's no mixing, these don't get homogenized, so they stay fixed in these different populations. We can then compare these and start to understand something about the relationship. So in this simple example where we're looking at 10 nucleotides, we have an 80% sequence similarity, so that tells us something about how closely related these organisms are. The more similar the sequences, the more closely related. The less sequence similarity, the more distantly related. We can even start to estimate times of divergence. We'll talk more about that in the next chapter. Now, we're never really using 10 nucleotides at a time. That's not very informative, but maybe hundreds or thousands of nucleotides, or nowadays even whole genomes, we're making comparisons. And we can take the example of ribosomal DNA with some representative organisms here and start to piece together who's most closely related to whom based on DNA sequence data of this homologous gene. That's critical, right? We can't take ribosomal RNA from one organism and then some other gene from another organism because those aren't homologous. We need to use homologous genes to make these comparisons. And here, humans and dogs are most closely related to one another. They're mammals. Salmons next, closely related to those two. They're all vertebrates, and so on. And we can take all these kinds of data and morphology, homologous structures, and start to put it all together into a really broad perspective. And there's a big project called Assembling the Tree of Life, trying to understand all these relationships between all living things and using these kinds of data. All right, another source of information when we're understanding this diversity and evolution of life on this planet is from fossils. So you probably all know what a fossil is, but it's preserved once living organism. A lot of times those organisms maybe don't exist anymore. They've gone extinct. Sometimes there's fossil remains of things that we still find today. And so the critical thing here is the organism gets buried in sediment, and then the calcium in bone or other hard structures will mineralize, so basically become rock. And then the surrounding sediment hardens around it and kind of preserves it and protects that. And then millions and millions of years, tens of millions of years later or longer, that structure, that sort of image, if you will, of what was a living thing is still there. Now, you might imagine, given this second bullet, calcium in bone or other hard tissue, we have lots of great fossils from organisms that have, say, an exoskeleton or bones, but soft-bodied organisms where they don't have structures that can fossilize very well, we have very poor fossil records of. Now, you've seen this slide before when we talked about Georges Cuvier, his sort of description and observations on how fossils form and how they are relative to one another in different layers. So here we have these different strata of sedimentary rock. They formed from this outflow of the river, this silting over time, layering. And over millions of years, we have older fossils in these lower levels and younger fossils in these upper levels. And so we can visualize this in a bunch of different ways. Here's an actual photograph of different strata. You can see these arrows pointing at these different layers. And we can see here in this cartoon, different fossils occurring over time. We see older rocks having these older forms, newer rocks having newer forms. And so the fossil record is pretty cool because this is a way to document the history of life through time. And we can see that here when we know, when do we start to see the first hominids, or when do we start to see the first vertebrate animals? When do we start to see land animals? We can find that information in the fossil record. It's just amazing. And we're learning new things all the time as new fossils are discovered. All right, so taking our understanding of how fossils form, we can make predictions, right? This is very useful in science. And so we say, well, if evolution is this gradual process, then gradualism over long periods, because we know the Earth is billions of years old now, does this fit? If we look at older layers of rock, do we find older things? Do we find intermediate forms in intermediate layers of rock? And the answer is yes. Yes, we do. And so we've created this theory, and we can put it to the test on a regular basis, and our data support it. All right, here's a question. We've got a diagram of some strata here, and there's an X here in layer B. So if this indicates the location of fossils of two closely related species in layer B here. So fossils of their most recent common ancestor, where would you find those? Which layer? All right. Yeah, you'd find them in layer C. So find that most recent common ancestor, you'd need to go into older layers, which would be further down, and you'd likely find that in C. I mean, maybe D as well, but the most likely possibility would be C. All right, let's take another fossil example. I just love this one with the evolution of whales. So we have modern toothed whales. They've been around for about 10 million years. But before that, we start to see in the fossil record things that don't quite look like whales. And an example here is this Rhodocetus here, this fossil that's from about 30 million years ago. It has whale-like features in its skull. It's got a tail here. But look, it has hind limbs. So it probably swam, maybe could get around a little bit on land. And here are some other examples. We're going back further in time in older fossils, 50 million years, 60 million years. We have Ambulocetus, ambulatory, like means able to walk around. But also this guy swims as well, Natans. And we go back a little bit further. Here's Pakicetus. There are features of the skull, and as we'll talk about in a moment, some other bones that are very much whale-like or animals related to whale-like in these organisms. And so we can start to then put this into the context of a family tree. And here, so we see modern whales here, and then we can piece together our fossils into these branches that dead end, right? These are organisms that don't exist anymore, and really put together this tree of life for cetaceans, these whales. Notice here on this top here, the hippopotamus. So this is the closest living relative of whales. And there's another piece of information I'll give you in a second. But first, a question. What part of this tree represents extant organisms? All right, yeah, it's the whales and the hippopotamus. I ask this because this is a term that students sometimes get mixed up with extinct. Extinct means they're gone extinct. But extant means they exist today. So these guys are the ones that are extant, the whales and the hippopotamus here. All right, so what's that connection back to, say, the hippopotamus? When we look at some of the these whale ancestor skeletons and bones, one of the things we note with fossil homology is this. So here's Pakicetus. Remember, this is the one potential whale ancestor from 60 million years ago. And if we look at these ankle bones, they have similar ankle bones to even-toed ungulates. And so the hippopotami would be in the same group, where we have these two humps on this bone. So here, like a pig or deer, or again, hippopotamus, versus other mammals that only have this one process, like the dog here. So many aspects of the fossil record can help us put the pieces together And then, say with the whales and the hippopotamus story, actually we have DNA evidence on those too, and that supports our estimates of phylogeny based on extant species. Getting DNA from fossils, that's a whole nother story. Now, I couldn't possibly skip over this one. Here's another one of my favorite transitional fossils. It's this guy here. This is Tiktaalik. And there's a great book called Your Inner Fish by Neil Shubin here that describes his discovery of this and a whole bunch of other things as well in that book. But the general story is this. Before 2006, what we had from the fossil record were lots of fish fossils that we knew were over 380 million years old. We have more recent fish fossils too. But before that, we don't see any real reptile fossils 380 million years ago or more. Then in about 365 million years ago, we start to see some primitive reptile fossils. So there's this gap of 15 million years or so ago that we can't really piece together. Where did we go from fish to reptile? And so Neil Shubin here, he was fascinated by this question, and he sought out rocks where in the world could you find rocks that fit within to this time frame, and they're accessible, and they might be fossil bearing. And so he found this in the Canadian Arctic, and he did a bunch of expeditions up there, and ultimately discovering the fossils of this organism called Tiktaalik, which dates back to about 375 million years ago. And so this is where we start to see a transition from fish to reptile. So some of the features that we see here that are worth paying attention to are the neck region of these organisms. So if we look at some of the fish fossils here, we see a fusion of head to body, whereas in Tiktaalik and then these later reptile fossils, we see the potential ability for these organisms to turn their head. So that's a critical feature. The other thing we see is forelimb evolution. So we go from something that's very fish-like, where we have these bones extending out and these rays of fins here. And then we start to see more bones in these different ancestral groups here. In the reptiles, we start to see things that really look like digits. In Tiktaalik, we have this intermediate type form where you've got these cluster of bones, which would be sort of like wrist bones, and then these smaller, skinnier bones out on the end, not quite fingers yet. And so this is very much a transitional form. But we do see the one bone, two bones, and then the cluster of little bones. So these are homologous structures that we see in our own, forelimbs, our arms. Something else we generally see, not only that ability, the limbs and the ability to turn the head, but we see a sort of a shift from laterally compressed heads in fish to dorsoventrally compressed heads in Tiktaalik and other terrestrial tetrapods. And so here's just a really cool figure showing that homology of the forelimb structure in these different organisms and us. One more example, if you can't get enough, and you can find so many of these fascinating examples. This is a story that came out a few years ago, but I love this. They called this the grandfather turtle, as this missing link in reptile evolution, where we have this reptile that has these very thick ribs. It's not quite a turtle yet, so this is a lineage of organisms that isn't quite a turtle, but we've got these bony structures on the abdomen, and then these ribs much more broad. So this was a really cool find, as they call this missing link in reptile evolution. Now remember, evolution isn't this stepwise linear thing, so that missing link term is very much a misnomer, but you will often see it in these popular news type articles. All right, our last piece of information here, our line of evidence here with evolution, is biogeography. Now, biogeography examines patterns of species distribution. And you can think of, well, what would influence that? Well, speciation, extinction, that's going to determine whether something's there or not. But then also geography from a huge scale at a continental level. or at an island level, islands form, right, and are worn away. Glaciation is really important. Islands are really cool examples, and we'll use some of those examples. And this is a great quote from David Quammen. Biogeography does more than ask which species and where, but also why. And this sort of starts to make that connection with adaptation, right? And how did something get there? But sometimes even more crucially, why not? So let's take a biogeography island example with our Galapagos Islands and these mockingbirds. Now, what we think has happened here in the Galapagos Islands, that these were colonized by mockingbirds from from South America, for example. And if that's what's happening, you would expect the islands to be colonized from east to west, because the eastern islands are closer to South America than the western islands. And if we look from a phylogenetic perspective, that actually is what we see, that these eastern islands were colonized, and then we have the separation of these other groups over time as they spread to these different islands. So islands are really cool for biogeography and contain many endemic species. And what endemic means is that that's the only place that they're found. And so if you're studying biodiversity, sometimes it's really cool to visit island chains, and it doesn't hurt if they're in the tropics either. But you're really interested in these endemic species, things you can only find there, and maybe you find a related species on a neighboring island. Now, where did these things come from? It would make sense that, South America is the closest landmass to the Galapagos Islands, and this kind of comes back to that question of, well, why or why not? How did these things get there, and why didn't other things get there? But there are other factors than just as the crow flies, if you will, proximity, or as the mockingbird flies in this case. Some genetic work was done, this is from 2006, on the mockingbirds in the Americas and in the Galapagos. And what was found, it's really hard to see in this tree here, but the take home was that the Galapagos were actually colonized from Central America, even though that's further away. And that might have to do with wind patterns or currents. There may be other explanations than just raw distance, although distance can be important. All right, so it's been observed that organisms on islands are different from, but closely related to, forms on a continent. What is this evidence for? Right, this is evidence of common ancestry. The islands might have been colonized from the continent, and that means that they have a common ancestor. But what's existing today, those are the organisms that are derived from that original ancestor, right? It's not that the one on the continent is currently the ancestor of the organisms on the island. They just share a common ancestry. So in biogeography, we can think broadly just of a couple of models of species distributions. One is the dispersal model, and this is like that island example. So a continental population that spreads to some islands here and then diverges over time. So we end up with these different populations isolated on these different islands. So there's a distance and time component. Another model here is called a vicariant model. In this example, you have some ancestral population that's split somehow. And this may be, as we'll see, think of like continental drift. We're talking about a split. Whereas dispersal, we're establishing new populations, spreading. Vicariance is splitting an existing population and then given some barrier to gene flow. So again, time and distance potentially, or something blocking this, you have divergence of these two populations. So continental drift, of course, is a great example of this. Here's what the Earth looked like about 225 million years ago. One continent here, Pangaea. And as over time, we start to see, say, South America and Africa split over 100 million years ago. But things that occurred on that continental landmass before this split, they would have had ranges that overlap both of these continents. And so we see that if we look in the fossil record here, we find organisms or fossils of organisms in both South America and Africa that overlap their historical range. And so there's many examples, plants as well. So here's fossils of this particular plant. You find Australia, Antarctica, India, Africa, South America. Those places are completely distinct now, but they used to be connected. Now, continental biogeography isn't all just about splitting up. Think of, we go back here, here's India, Indian subcontinent, which eventually plows into Asia here. So you have land masses coming together. Another cool example is in the Americas. So only about 3 million years ago or so did the Isthmus of Panama form and connected North and South America. Previously, they were distinct, separated here, but now they're connected by this Isthmus of Panama. And so what happens is you have organisms that were in South America or North America moving north or south and spreading to these new places. So we see that why or why not, where do we find species changing in different ways depending on some of these geographic factors. All right, that's it for Chapter 19. We'll see you next time.